Imagine a world where a simple scraped knee could lead to serious complications or even death, or when life expectancy was far lower. What appears to be a simple cold might actually turn out to be something far worse. This was the case before the advent of antibiotics, which have revolutionized the way we address disease. It would probably be fair to say that no single type of drug has influenced human health more than antibiotics. These miracle drugs have extended life and made most infections in general a simple inconvenience. But what exactly were in those pills? How did it work? What are we putting down our throats? For a closer look, we should start by defining what an antibiotic is. Antibiotics are compounds which have a toxic effect towards microbes such as bacteria and to a lesser extent fungi. If we look at the tree of life that taxonomists use to classify life, bacteria would be in a separate branch from where humans are classified. Part of what makes antibiotics special is the fact that they are more toxic to these microbes than they are to our own cell. This is an important criteria in drugs known as selectivity. One of the ways of classifying these antibiotics is by mode of action, or how the compound is actually toxic towards bacteria. There are many antibacterials which have been developed, and one could easily spend years learning about the lengthy history of these molecules. But in this brief survey, we will focus on general groups of these drugs. The important thing to remember is that, whatever the class, the end goal is the same, which is to stop harmful bacteria from proliferating in humans. Three general classes of antibiotics include cell wall inhibitors, enzyme inhibitors, and protein synthesis inhibitors. There are others, but most of the ones you'll encounter will fall into these general groups. With this episode, we will first focus on cell wall inhibitors, what they look like, and how they work, since they provide a nice introduction to antibiotics in general. Later, we will cover other classes and start to look at why some have stopped being useful and what is currently being done to fix this. Cell wall inhibitors are some of the most common antibiotics used. This is in part because penicillin, the grandfather of modern antibiotics, is in this class. And while there were antibiotics that came before penicillin, it is hard to argue that one has had as large of an effect on society. The fact that it is still used today is a testament to this importance. Bacteria have an outer coating that's known as a peptidoglycan cell wall, named so because it contains both peptides and sugars in repeating units. This layering can essentially be thought of as an armor for the cells, protecting them from threats. Part of what makes the peptidoglycan so strong has to do with its cross-linking, which takes long strands and connects them together. Here, the strings of sugars are shown as large circles, and the smaller circles are peptides providing the linkages between the chains. The difference between cross-linked and non-cross-linked is similar to that of fabric. Individual threads all lined up in the same direction do not hold together well, but once they are interwoven, the resulting material is much stronger. This cell wall is maintained by proteins, called enzymes, which are constantly dismantling, reassembling, and rebuilding the peptidoglycan layer. The enzymes that cross-link the peptidoglycan look for special points on the layer which they can stitch together. Penicillin and related antibiotics inhibit this enzyme by getting caught in the enzyme like a wrench getting stuck in the gears of a machine. But how does penicillin do this? The answer lies in the structure of the molecule and how similar it appears to the marker which the enzyme looks for. Because they look so similar, the enzyme will mistake penicillin for the marker and get stuck. This effectively shuts down the enzymes that stitch the crosslinks in the peptidoglycan. When the bacteria cannot cross-link the cell wall, several things happen. First, the cell wall now has holes through which other molecules can enter. Like a gate that is missing parts, unwanted molecules can now pass through. Next, if the bacteria tries to divide to make more bacteria, it will not be able to make more cell wall material, and will wind up shedding the peptidoglycan and exposing itself. Like a water balloon, the cell wall stops the insides of the bacteria from spreading outward, but a hole can allow this inside material to ooze out. Penicillin now has many variants which chemists have made that change small parts of the molecule to help it perform better. One might imagine that a molecule which fits better into the enzyme would do a better job of stopping the machine from stitching the peptidoglycan together. Shown here are some of the variations that have been made, but notice that in all the changes, the critical center of the molecule stays the same. This concludes the first installment of our series on antibiotics. As always, comments and questions are encouraged, and we hope that this has been an interesting and accessible look at common chemistry.